Thank you, Billy. So just briefly to introduce myself, my name's Andy Cowell. I am an associate professor in the Department of Medicine at Washington University School of Medicine, and that's in St. Louis. So for me, it's 8 a.m. It's also election day. So uh, it will probably be a very exciting day for all of us. So with that, I'll go ahead and kick things off. So to give you a little bit of background about my lab, we're interested in microbe-immune interactions important for airway health in particular. And so most of this talk is actually coming from, at least from my perspective, from the microbiology perspective. And so let me just make sure this is working. Okay, so to give you a little bit of background on microbial ecology, and I know we've already gotten into this topic some, but I just want to make sure we're all on the same page here. I think there's been a growing appreciation that microbes can influence human health in really diverse and sometimes surprising ways. And as Matteo just talked about, we know that microbes are intimately linked to metabolism in human hosts, but they can also influence the immune state in the host. So there's really good evidence that microbes influence the manifestations of inflammatory bowel disease. There's evidence that they can also influence diseases like asthma and autoimmune diseases. And many of these studies have used very sophisticated microbiome profiling techniques to basically look at the composition and functions of the gut microbiota, and they find these very strong signals of something about the gut microbiota that correlates with either disease presence or absence, activity, or severity. The problem is that using the gut microbiome as a biomarker is really challenging, and I think you guys know why, right? This is a breath biomarker conference, so I think that I'm preaching to the choir, so to speak. It's fecal samples in general are difficult to collect. They have to be patient collected typically. And they have to undergo a lot of processing in order to translate that sample into data that we can then interpret and make a prediction about disease, right? And this makes it actually a major barrier to translating some of the diagnostic and functional things that we've discovered about the gut microbiome to clinical practice. And so this project for us began, make sure my slides are going forward here, when my colleague, Audrey Odom-John, who is going to be speaking a little bit later today, had published this really beautiful paper suggesting that the malaria parasite, so an infectious agent, produced terpenes that could be detected in human breath, and that could be used as a diagnostic biomarker for people that were infected with malaria. And so we began this project to explore the idea that colonizing microbes, especially those in the gut, could influence the VOC composition that we find in breath, so the volatile organic compounds that we find in breath. And if this is true, this opens up a lot of possibilities that I think we can all appreciate. So that means that if the gut microbiota composition is reflected in breath VOC composition, this means that we can use VOCs to really get a look into the gut. And this allows us then to now begin to use breath VOCs as a biomarker as a marker for the state of the gut microbiome. And so this is a much more accessible window into the gut microbiome than, say, processing fecal samples, performing metagenomic sequencing, and so forth. And so this project was mostly led by my graduate student, Ariel Hernandez-Leyva, as well as Amalia Berna from Audrey's lab. So why is this a feasible possibility? And again, I think that this has been covered already to some extent, but I think when I think about the gut microbiome, one of the most striking properties that we have learned about it is that it provides this incredible set of dining utensils, if you will. This is a kind of one of the analogies I've heard a lot, but the gut microbiota provides this set of dining utensils that extends our base metabolism and allows us to metabolize things like fibers and other undigestible food products and break them down. But as a product of this microbial metabolism, microbes make all sorts of small molecules that can be found throughout the body. And I'm highlighting a study here published a few years ago showing that metabolites produced by microbes in the gut can rapidly transition to the entire body. They can be found in the brain, the liver, the pancreas, everywhere. This idea that microbial metabolites distribute throughout the entire body suggests that if we look in the breath, we may be able to find metabolites that originally came from the gut. Likewise, I think that VOCs can actually give us taxonomic information about the microbes in the gut. And this is a really nice paper that was in Systems a few years ago that shows that the types of volatile organic compounds produced by different microbes actually corresponds to their taxonomic relationship. In other words, the types of VOCs the microbes produce tends to be similar the closer these microbes are related to one another. And I think for a microbiologist, this makes 100% sense, right? Most of us have worked with E. coli, and we all know that E. coli has a distinct smell. And that's actually true for different microorganisms. That, you can often smell the type of bacteria that you're working with just by the odor, suggesting that these volatile organic compounds are actually different in between these organisms. And there may be relationships of the types of compounds that are made with the taxonomic and evolutionary relationships between these microorganisms. So the story I'm going to tell you about today is we've actually released on medRxiv a couple months ago. It's there, so if you have questions, please take a look at our manuscript. We cover all of this in much more detail there. Okay, so we actually began with a human study, and this was a fairly small study where we had looked at kids aged 6 to 12 that were healthy. So there was nothing wrong with these kids. And we asked them to refrain from eating or brushing their teeth for a couple hours before we collected breath from them. We also collected a stool sample at approximately the same time. It's hard to get those to line up exactly, but all the stool samples were collected within in a fairly short time frame around that breath collection. And our objective was to determine if the gut microbiota composition or functions correlated to the types of volatile organic compounds that we saw in breath. And so we performed this study. We analyzed the breath samples by GCMS, and we performed shotgun metagenomic sequencing on the stool samples to look at compositional differences as well as functional differences within the gut microbiomes. And there's a ton of analysis that we did on these samples, but I'm going to give you an overview of them just to, and for the sake of time. And so what I'm showing you here are principal coordinate analyses. So most of us are familiar with principal coordinate analyses, but each of these circles represents an individual. And the closer the circles are to one another, the more similar the gut microbiomes are to one another. Additionally, we are superimposing the breath volatile organic compound profile on top of this. And so if you take an individual and you follow the arrow, at the end of that arrow is where on a principal coordinate analysis, those VOCs would belong. And so what we're doing here is we're performing a Procrustes analysis. We're actually trying to align the volatile organic compound data, breath data, to the microbiome data. And we're trying to do this in such a way that really maximizes their correlation one to another. And this is basically a very sophisticated statistical tool that allows us to correlate how similar and how well these two different data sets correspond to one another. And just like any other correlation, the protest value, which is our statistical value here, it goes from 0 to 1. Closer to 1 it is, the higher the correlation. And so when we look at the composition, so the species that are present in the gut microbiomes, and we correlate that to breath VOC data, we see a fairly strong correlation in between gut microbiota composition and the breath VOCs. Likewise, if we look, instead of looking at species, we look at the functions that are present. So the orthologs, the protein orthologs that are present in the gut microbiome, we actually improve that correlation a little bit. And again, we see a correlation in between breath VOC composition and microbiome composition. And finally, when we start binning those functions into pathways, and we use the pathway abundance data and correlate that to breath VOCs, we again see that there's a correlation in between breath VOC composition and the presence of these pathways. Again, suggesting that there's a correspondence of gut microbiome composition and function to breath VOC composition. So we dug a little bit deeper here, and we wanted to look at specific compounds. So we first identified a set of compounds that we felt, based on the literature, that we felt were reliably found in human breath. And we performed a supervised learning algorithm called random forest to basically estimate how different microbes correlated or corresponded to the presence of those compounds in breath. And I'm showing you the summary of the results over here on the left. And what this is showing is that a very large percentage of the variance can be explained by the types of microbes that are present in breath. And so, for example, for example, caffeine, up to 40% or more of its variance within our data set can be explained by the species present in the gut microbiota. And most of the compounds that we looked at we could explain 20 to 50% of their variance in our data set. And so when we wanted to look at specific taxa and specific taxa that corresponded to specific VOCs in the breath, we've highlighted a few here, but one of them that I think makes a lot of sense and kind of dovetails with what we just heard about is we found that acetic acid, which we found in breath, had a correspondence with Bacteroides vivatus. And so the way to interpret this is that things that are more yellow, the taxa is present in higher abundance, and the higher this value is the how well that feature, and in this case, Bacteroides vivatus, explains the presence of acetic acid. And we thought this was very satisfying because it turns out that Bacteroides vivatus is a known short-chain fatty acid producer, and short-chain fatty acids include things like acetic acid. And so the fact that this bacteria corresponded well with the presence of acetic acid in the breath made some sense. Of course, what I've shown you so far is all been from human data, and it's retrospective. And we really wanted to test this idea in a more rigorous way and prospectively use this data, hypothesis to test whether or not the composition of the gut microbiota could influence breath VOCs. And for this, we needed an animal model. And so we turned to mice, our favorite animal model. And we happened to have access, because of my lab's interest in respiratory physiology. We had purchased this FlexEvent, which is basically a very sophisticated machine to measure pulmonary function in mice. But one of the nice features about this instrument is that it allows you to basically collect defined air on one end of this machine. So we can use clean VOC-free air, or at least reduced VOC air, and pump it through these mice. And then on the opposite end, we can attach a bag and capture the exhaled breath from these mice. And an additional benefit of using the system is that it uses a tracheostomy tube. So we're bypassing the oral microbiome and the oral pharynx altogether. And so to test this system, we injected mice with isoprene over a period of a couple weeks. And as we know, isoprene is a quintessential breath VOC, but not really abundant in rodents, it turns out. So it was a very good VOC for us to look at as a positive control. And so we injected mice with either isoprene or a vehicle control, and then collected their breath after 2 weeks. And what we saw was that mice injected with vehicle alone had very low levels of isoprene in their breath, whereas mice that were injected with isoprene had higher levels of isoprene in their breath. Okay. So this set up an experiment where we could start now to test how gut microbiota composition influenced breath VOCs. And to briefly summarize a pretty complicated experiment, we had two groups of mice, or actually three groups of mice. We had a group of mice that came from a vendor called Taconic, and we had a group of mice that came from another vendor called Jackson Labs. It's known that the gut microbiota composition in between these two different vendors is distinct from one another. So we know that the gut microbiota in between these two groups of mice is different. We had a third group of mice which were germ-free and remained germ-free throughout the entire experiment. Okay, we collected breath from each of these mice. After we collected breath from them, we then took the microbiomes from these each of these groups of mice, so from either the Taconic mice or the Jackson Lab mice, and we transplanted them into germ-free mice. So now we've made a conventionalized mice. So conventional mice are the Taconic and Jackson. And then the former germ-free mice are the conventionalized mice. And those conventionalized mice receive the gut microbiota from either Taconic or Jackson Lab. So we're basically giving them back microbes. And we collected breath from all of these groups of mice about 1 week after colonization. And again, I'm just going to summarize the results here. This was, again, a fairly complicated experiment, but I am, again, showing you the Procrustes analysis down here. Again, the points represent a mouse, and the arrows represent the VOCs in those mice. And what we're looking for is for the arrows to be short and to point to different distinct areas on this principal coordinate analysis. And again, using this Procrustes analysis, we can see that the composition and the functions of the gut microbiome correlated to the VOCs that we were recovering from breath of these mice. And an additional analysis that I'm not showing you here, we can also show that each distinct microbiota composition corresponded to a distinct VOC composition in these mice as well, suggesting that the transplantation of these microbes is changing the VOCs in these mice and leading to compositionally distinct VOCs recovered from breath. So this was really cool, right? And at least we were very excited by this idea, but we are working with complete microbial communities. In other words, there's an unknown number of microbes present in the gut microbiomes of these mice from Jackson or Taconic labs. And so as a community, they may have functions that are not present in single microbes. And we wanted to ask the question, if we put a single microbe in a mouse, will it change the VOC profile? And if we know that a single VOC is produced by a single microbe, can we demonstrate a direct contribution of that microbe to the VOC composition in a mouse? And so to do this, we had to develop a method to collect VOCs from an anaerobic microbe. And so what I'm showing you here is our setup. So in the Erlenmeyer flask over here on the left, you're seeing basically a microbe that has been grown for a day or two under strict anaerobic conditions. And we are then collecting it to a stopcock that there were two different stopcocks, one of which is connected to a bag filled with anaerobic air, and the other is connected to a pump and a thermal desorption tube, so a collection tube. And so what this allowed us to do is then pump that anaerobic air through the headspace of the anaerobic culture, and then collect the VOCs onto the TD tubes for GC-GCMS analysis. And we did this for 5 different microbes to start with, just to test the idea, are we going to see VOCs that we recognize that are found in human breath? And this is a summary of the results. I'm showing you basically all of the VOCs that we found. But I just want to highlight that we saw that, first of all, almost all the microbes, basically all the microbes produced VOCs that were different from control. So there were VOCs that were enriched in each one of these cultures. But one of the things that was interesting is that we did see that there were a lot of microbes that produced similar VOCs, or the same VOCs. Like, for example, isoamyl alcohol was associated with multiple different bacteria. And a lot of these VOCs that we're seeing produced by anaerobic bacteria are VOCs we recognize from breath and that have come up again and again in breath studies, such as toluene and benzene and nonane and many others. So this was encouraging because it indicates that these microbes are making VOCs that are potentially things that we also see in breath. So this set up the experiment that we were really excited about, which was, now we've done the monoculture, and we profiled the headspace from those monocultures and demonstrated that they make VOCs. The question is, if we take those microbes and we put them into mice, can we recover VOCs from that mouse, right? So there's a former germ-free mouse that we're now going to colonize with a single microbe. Does that colonization result in production of new VOCs in that mice, and are those VOCs similar to what we saw in monoculture? And so we did this for each of the 5 microbes I showed you. We colonized mice with them. We allowed them to hang out for a couple of weeks, and then we collected breath from those mice and profiled them here. And again, I think one of the things we can conclude here is that colonization with a single microbe changes the breath VOC composition in these mice compared to germ-free controls. And we see enrichment of a number of VOCs that, again, have come up many times. So for example, we see that mice colonized with B theta, so Bacteroides theta iota omicron, E. coli, as well as colon cell aerophoresians seem to make more toluene than germ-free controls in their breath. And so again, we're seeing that monocolonization is altering the breath VOC composition. And so we wanted to compare this to the culture data, which I'm summarizing here. So on the x-axis, we have the change in log-fold change from media. So media control versus monoculture. And on the y-axis, we have log-fold change from germ-free mice. And over here in the upper right are VOCs that we are finding both in the headspace of culture as well as in the exhaled breath of a monocolonized mice. And they include things like ethyl acetate, which is produced by E. coli both in vitro as well as in monocolonized mice, and benzothiazole, also produced by E. coli. And we also saw things like toluene, which is produced by colon cell aerophasians in culture as well as in monocolonized mice. Okay, so I think I'm out of time here, so I'm going to go ahead and wrap things up. I've shown you that breath VOC composition correlates to the gut microbiota composition in kids. We've also, I showed you our method for colonizing and capturing VOCs from mice, as well as testing the influence of different microbiota on breath VOC composition. And I've shown you that that VOCs that are captured in monoculture can also be recovered from mice monocolonized with that same microbe, at least for a couple of the microbes that we've looked at so far. So one of the things that we've gotten very fascinated in is trying to trace the origins of these microbial of these VOCs. And the hypothesis that we entered this project to test was, do VOCs produced by gut microbes directly contribute to VOC composition and exhaled breath? And I think we have some evidence now that supports that idea that some of these VOCs are making their way all the way from the gut to the exhaled breath. But while we've been thinking about this and testing these ideas, I think it's occurred to us that there's probably a variety of mechanisms by which gut microbes can influence breath VOC composition. So there is this direct contribution method, but there's also perhaps a role for gut microbes in providing substrates for host metabolism, and those host metabolites are actually what shows up in breath. So you can imagine that this is probably a major mechanism by which gut microbes contribute to breath VOCs. Additionally, as we just heard about in the prior talk, it's possible that host metabolism is altered by gut microbiota composition, and there's a lot of data suggesting that happens in a lot of different diseases like obesity and malnutrition. And then finally, one that we've begun to think about a little bit more is the role of gut microbes in modifying absorption of dietary molecules. And this is a well-known function of the gut microbiota to modulate gut permeability. It's likely that the modulation of gut permeability is changing the absorption of compounds that can partition to breath. So we have a variety of next steps. We're performing some additional clinical studies to look at individuals with rapidly changing gut microbiotas. And we're also really trying to dive deeper and identify specific microbial pathways that lead to specific VOCs in breath. So with that, I need to thank the Odom John Lab at the Children's Hospital Philadelphia, especially Audrey Odom John, Amalia Berna, Samantha Whiteside, and Young Liu, as well as my lab who actually did all the real work here. And I'll take any questions. Thank you.
Thank you so much, Andy, for a really fantastic talk. I was hoping to kick off with two quick questions I had. So when you were looking at the compounds on breath for the human subjects, so you talked about acetic acid and then how that relates back to the microbiome. Were there other short-chain fatty acids that were detected, or were they not something that you were picking up with the analytical method? You'd expect to see, butyric acid and other fatty acid compounds as well.
That's a good question. We'd have to go back and look at the data. We intentionally reduced the number of compounds we looked at so that we have an N problem when we are trying to look at all compounds that we are detecting by GCMS. And so when we tried to do analyses corresponding gut microbiota composition to VOCs, we found that it was the best way to do this was to actually just focus our question on a select set of VOCs we felt were reliably found on breath. So we made some sacrifices to do that, and one of those is that we're not looking necessarily at all short chain fatty acids that may be there.
Got it. Thank you. I was really fascinated by benzene and toluene. So when you showed the benzene on the human study to start with, so I had always assumed that it was just environmental exposure. But when you're saying in the mouse and then transplanting into the mice that single microbe as well, it does seem quite striking that you are getting increases in benzene and toluene there. I was wondering if you could elaborate a little bit further on the mechanism for that. Why do those compounds kind of appear upregulated?
I wish I knew. Like I don't, and this has puzzled me since we've been looking at this data. Obviously benzene isn't a friendly compound. Neither is toluene or xylene or any of these other compounds, and yet they show up in other human breath studies. And yes, I've seen that the thought is they're coming from environmental sources, but we've been very careful to control for that as well as we can. It is possible they're coming from the environment in our experiments. I'm not sure how or where, because the human study was actually performed at a clinical site, so is different and is not going through our physical lab space where we're doing these culture studies. And so yeah, and I think that we have consistently seen that there is production of these VOC of xylene and toluene and these others in microbial culture. It's hard to know with absolute certainty that it's not environmental, right? These are incredibly sensitive assays. So yet we have consistent, and we compared to controls, we find that they're enriched in our microbial culture. I don't know how. And I think that is like one of the open questions, is that these are really strange compounds, and I'm sure that if they're being made, what I'd love to do is really go back into bacteria and really define how those VOCs are being made. And
and as you say, it would show up in the controls as well. So that's why it seems so compelling that you do have genuine production of it. That's really fascinating, really fascinating. And then one last question for me, and then we'll hand it over. So you talked a little bit about fecal microbiome tests and some of the challenges there. It was kind of interesting what you're saying, that you can have multiple bacteria that lead to the production of single VOCs. So I'm thinking about where you do have fecal microbiome tests that are telling you about the presence of specific bugs. Do you think breath has the potential to replace a fecal test, or is it more of a complement where you're looking at the activity of the microbiome, because you're getting a direct measure of the end product?
I think that they can be deployed in very specific. So in fact, this is already a test, right? The hydrogen breath test is something that's already commonly used, and I could certainly envision that a similar type of system could be used to look for specific functions of the gut microbiome in a very sensitive way, where specific compounds are administered to somebody, and breath is collected from that individual afterwards to look for metabolic products. And I think that assays like that are probably going to be more robust than just looking at gut microbiota composition, which when we're looking at fecal samples, and this came up in the prior talk, when we're looking at fecal samples, we're looking at the microbiome that's present in the distal gut. Yeah. And that has limitations, right? And whereas when we start to look at functional things, like going back to the hydrogen breath test, we're now looking at microbes that are present higher up in the gut, which is in a lot of ways a lot more useful than just looking at things that are in the feces. So yes, I mean, I think there could be a role for these for these tests that are very specific and clinically helpful information. Yeah.
Fantastic. Next question is, do you think these VOC results could change as bacteria evolve, i.e. with antibiotic resistance, other differences per strain, or do you think there'll be some core VOC signatures?
Well, that's a good question. I think there's probably going to be a little bit of both, right? So I think the core VOC signatures, and I think we see hints of that in the data, and that we're seeing VOCs that go up in the presence of a single microbe, but across different taxonomically very distinct microbes. And so I do think there is probably a core set of VOCs that are there. Now, whether they're directly contributed or are generated by another mechanism, the host, we don't know yet. But I think there are probably going to be these core metabolites that are there, but I do think that there are going to be metabolites that are very or somewhat specific to the taxa, and that comes from other data that's been published where there's specific VOCs that are associated with specific microbes. Yeah, and I think one that's already come up in these talks are things like indole, which is reflecting a very specific microbial function, the tryptophanase. That is present in many Bacteroides. And yeah.
Excellent. Next question. Wonderful initiative towards understanding the origin of expelled human VOCs. What type of media was used for the anaerobic bacterial culture experiments? Now, are you planning to use blood and/or urine-based biomarkers alongside fecal and breath VOCs in the future to study the role of microbial contribution to host metabolism?
Good question. So we used a specific media, which is basically a standardized media in my lab, where we take a standard, I think it's BHI media that we supplement with mucin. So it's a porcine gastric mucin that helps a lot of these strict anaerobes grow robustly. So that's the media. And yes, I guess leading to that question, do we think the media influences the VOC composition? Absolutely. Yes, absolutely. Troublingly, a lot of these gut microbes don't have defined media under which they will grow. So we have to use these kind of undefined media to get robust growth for them. But we do, all these experiments were performed with the exact same media over time. So we don't think that it's small batch differences in media that's leading to this difference. And again, we were specifically using media and using the same media for all these bacteria, so we don't think it's due to that. Yeah.
Got it.
And then the second part of that question was, I'm sorry, remind me.
Are you planning to use blood and/or urine-based biomarkers?
Oh yeah. Not at the moment. It's a great thought, and we certainly have some interest in that direction, but we have not planned any studies using these other sources yet.
Got it. Time for one very quick question. Have you done or plan to do any work to detect sepsis in children, particularly in young babies?
Not sepsis particular, but we are very interested, again, looking at states where the gut microbiome is rapidly changing, and that includes antibiotic treatment as well as early childhood where kids are acquiring microbes over time. So yeah, not sepsis in particular, but that is certainly one of the eventual applications for something like this. Yes.
Excellent. Well, we'll leave it there. Thank you so much for a really wonderful talk. Really appreciate the time to walk through the results and answer the questions. And I love that. No problem. I love that point that you can smell the bacteria that you're working on in the lab.